My name is Megan Rokop, and I am the director of the Broad Institute's Educational Outreach Program. I'm pleased to introduce our speaker tonight, Dr. Pardis Sabeti. Pardis is currently a postdoctoral researcher here at the Broad Institute, and her work focuses on studying variation between members of a population. And as I'm sure that she will tell you tonight, sometimes the population that she's studying is a human population, and other times it's a population of the parasite Plasmodium that's the causative agent of malaria. Pardis received her undergraduate degree here at MIT and her master's and her doctorate in biological anthropology at Oxford, and she received her MD at Harvard Medical School. In the fall, Pardis will be beginning as an assistant professor at Harvard University. And please join me to welcome Pardis Sabeti.
Thank you. Can everyone hear me all right? Is that okay? Well, thanks for having me come out here. This is an interesting exercise. I haven't done a lot of sort of public consumption of science. So you can always do votes from the audience if it's not clear, if it is clear. So what I'm doing, I'm trying to make it jargon-free, but sort of take you on a little bit of give you a background of how we study natural selection first in humans, and also how we can apply those same types of tools to study selection in pathogens. So first I want to kind of give you a background on the study of natural selection. So bear with me. We've been thinking about natural selection for a very long time, about evolution for a long time. Even before Darwin and Wallace, there was Lamarck, there was Huxley, Lyell, Malthus. Many people thinking about the idea that species evolve and adapt to their changing environment. But it wasn't until 1858 when Darwin and Wallace proposed in a set of 2 papers the idea of natural selection, a hypothesis by which evolution happens. And it really set the dialogue going on thinking about evolution. And in 1871, Darwin published The Descent of Man that really brought humans into the forefront. So the idea that not just other species on Earth, but humans themselves are subject to evolution. It was actually not until 70 years later, in 1948, though, that we had any real example of human evolution. So J.B.S. Haldane, who was one of the founders of population genetics, he made an observation of the natural world. He looked around and he noticed that there are many, many red blood cell disorders, like thalassemias and sickle cell. And he noticed that they all seem to congregate in tropical areas, areas where malaria was prevalent. And so what he proposed was something called the malaria hypothesis, which is that the reason that there were so many red blood cell disorders in these tropical regions is because they may be protected from malaria through the process of evolution. And in fact, actually, just a few years later, Allison and others showed that, in fact, the sickle cell trait is always present in malaria endemic regions, and individuals who carry the sickle trait are protected from malaria. So that was, like I said, 70 years later, the first example of human evolution. And since that time, there have been very few examples shown, actually. So we can think of many different things that drive human evolution. We can think of pathogens, changing environment, changing temperatures, domestication of plants and animals, and other cultural changes, and also the need to constantly reproduce and have and bear many children. But in the 1950s, there was the sickle trait at a gene called hemoglobin that was found to be under selection. And that was, there wasn't much more that was happening in the last 50 years. And that's because it was very hard to generate the data by which to do that kind of analysis. Probably the best example didn't happen until 50 years later, which is lactose tolerance. And I'll tell you more about that later in the talk. But is the ability to drink milk is something that was, as an adult, is something that appeared to be under strong evolutionary pressure. And so now, though, the transition has occurred where we have data from the whole genome. We can study genetic variation throughout human populations. In 2004, that became possible with a series of projects studying genetic variation in the whole genome. And once you do that, you can find those kinds of signals that we find at sickle cell and hemoglobin and lactase. We actually can see those same types of signals throughout the genome. And but now the challenge turns. from before we had candidates that we're looking at, that we were identifying selection at. Now we have regions of the genome under selection, and we have no idea what's driving that. So we're at this new phase where the challenge is to find out what is underlying these events. And so in the talk, I'm going to kind of do three things. I'm going to talk to you about the method by which I study selection most prominently, sort of that's my area. Then I'll talk about natural selection broadly in humans and what we've found so far. And finally, I'm going to talk about natural selection in one of the pathogens that affects us, Plasmodium falciparum malaria. Okay. So this first thing, here's the, this is the public consumption. The first thing is this term long-range haplotype, and hopefully I'll explain to you what that means. But it's a method by which we can study recent selection in humans. So as I said before, everything that we sort of, so the kind of the purpose by looking at natural selection is a very fundamental principle set forth by Darwin and Wallace. It's a very simple principle, and most everything that I talk about today is based on this very simple principle. And it's the idea that take a population of individuals. So here's a population of individuals at one generation. When a new trait emerges, one that is somehow beneficial to the survival or the reproductive success of its carriers, it'll spread quickly through the population. So that trait is beneficial. Those individuals who have it are more likely to survive and reproduce, and so the trait will spread through the population. And that's in comparison to the rest of the genome, which we don't think is actually driving these sort of physical traits that are under selection. So those things will just emerge in the population, and they'll drift around. Their prevalence sort of doesn't change that dramatically generation to generation. So if they're around long enough, they may drift up to a high prevalence, but that takes a very long time. So if the selective event, if the pressure happened recently enough in time, we can actually measure the prevalence of the variant, compare it to its age, and look for a disparity. So what we're looking for when we look for natural selection in recent past is something that is much too common to have gotten to the prevalence that it's at in such a short period of time. Okay. And so the prevalence is something easy to measure. You just sort of find out, take a population of individuals and find out how many individuals carry a particular trait. What's more difficult is actually the age. How do we determine how long something's been around? So there's a sort of a principle by which we can look at this, and it's a correlation. Now here, this is where you bear with me. So when a variant emerges, it occurs through something called a mutation event, where it'll happen on a chromosome. A mutation will occur, generating a new variant. When that mutation originally occurs, it happens on a chromosome in an individual. And so that variant is originally correlated with every other variant on the chromosome of origin. We call that a haplotype. Every generation, there's a reshuffling process that happens called recombination. And so generation after generation, as that variant is around, those correlations are going to be broken down by this reshuffling called recombination. And so the take-home message is that young variants will lie on long haplotypes, and old variants will lie on short haplotypes. And so based on that, what you say is, absent selection in a neutral environment, young variants will lie on those long haplotypes, and because it takes a very long time for them to rise in prevalence, they will be at low prevalence. Old variants will have the short haplotypes because of all the reshuffling that's taken place, but they'll have either low or high prevalence. They've just drifted around to some prevalence in the population. And when we look for positive selection, this driving force is a young variant, which we can determine by the long haplotype, and the high prevalence suggesting that it's under selective pressure. And so I just want to shift around and show you all the different ways that we can look at this. So now basically it's still always looking at prevalence to age and looking for this kind of disparity. So this is what, when we plot out that, when we look at the age versus prevalence, this is what variants in the human genome might look like, is that things that are at low prevalence, or things that are young will be at low prevalence in the population because they haven't had a lot of time to go to high prevalence, but they lie on long haplotypes. And that things that are old can be at any prevalence in the population because a lot of time has passed and they drift around, but they'll lie on short haplotypes. And the things that we look at are out here, positive selection, something with a long haplotype and a high prevalence. Okay? So that's the background. And now we'll start looking at how we look at that data and how do we actually measure that. So let's say there's a particular position in the genome, a particular variation that exists in the genome, and you're interested in it. So let's say it's an AG variation, a nucleotide of A that moves to a G. Well, we can measure the prevalence in the population, and then we can look at all the variations that exist on the chromosome, like looking at the long-range variation across the chromosome, and we look for that correlation, and we look how long it lasts. And the way we're measuring it is, let's say, particularly, so you're interested in the G variant, okay, that form of that. For that variation, it can be either A or G, and you can look at the G variant. So when that mutation occurred generating the G, it originally did it on a chromosome. And there's all these other variations across on the chromosome, but let's say the individual chromosome it occurred on was something like this. So CAG-TTC at those positions at far distances. Well, see, you can see here that these are all variable markers. So you could either be a C or a T or an A or G at each of these positions, and there are other individuals in the population that have those chromosomes. The reshuffling starts to happen. Through recombination, essentially the background on which that G lives begins to decay. And so there are many, many other backgrounds that it can exist on. And so how I measure the length of haplotype, this correlation, is by something called EHH, extended haplotype homozygosity. But the idea of it is just a simple mathematical measure, which is given that you have the G, so we're interested in people who are G at this position, what's the probability that at this position out, any two individuals, any two G individuals have the same variant? Well, everybody at that position is C, so it's 100%. But then you say at this position out, what's the probability that if you randomly took two individuals that carry that G variant, what's the probability that this distance out they're the same? Well, there's a little bit of breakdown. So at this point, it's 53%. And as you go further and further, it decays. But we can measure that process of decay and look at it, and that's how we measure this correlation. And so we thought about this and the ways of measuring that. And once we did, we applied it to two variants that we knew that we were interested in that protect from malaria. Because we can think that we have a sense that malaria is driving evolution because so many people die from it that if you have some sort of a variant that protects you from getting it, you're more likely to survive and reproduce. So we knew these variants are protected from malaria, and G6P has a well-known malaria resistance variant. And we looked at it, and here's the resistance variant. Here's a non-selected variant in that same locus. And you can see here that there's very little decay that's occurred at this long distance, right? So some decay has occurred, time has passed, but still there's a conservation of the original variant, the chromosome that it existed on. And that's compared to here where there's a lot more decay and a lot more time has passed. And we can measure that, we can plot that out the way that I showed you, length of haplotype versus prevalence, and we can see that G6PD stands out from what we would expect based on, at this point, theoretical simulations of what we'd expect. And we also, having found that, we then applied that to another gene where we found a novel resistance locus to malaria at CD40 ligand. We found the same pattern. So what we were seeing here is that these things that we had found a method by which we could look at genetic regions looking for selection, and we had seen is that many of the things that we'd been studying for malaria were in fact under beneficial selection. And once we did that, we started to apply it to other candidates. At this time, we didn't have, it was very difficult. We had to go candidate by candidate, looking at things that we thought were interesting. So one of the ones that we looked at first was lactose tolerance, and people had suspected that lactose tolerance was under selection. And so I'll explain that because it's not intuitive that in fact, actually, ancestrally, we were lactose intolerant, right? And that is that all mammals drink milk, but at a certain point, they wean. So most mammal adults don't drink milk. And there's a very clear reason for that. I mean, so for one, your mother does not want you to drink milk into adulthood. She doesn't want to give it to you until it's a socially awkward situation. And it's also that, but she wants to conserve her resources, right? She wants you to wean off of it and go and get other things, fruits and berries, whatever, so that she can conserve her resources. But once cows became available, that pressure changed because now it doesn't have to be your mom that gives you the milk. You can take it from somewhere else. So there was a selective pressure that changed that made it possible for us to switch the way that we express that gene to express it all the way. So not just in the first few years of life and not just to the weaning point, but on. And so we can see in Europe, one of the places where domestication of cattle happened earliest and milk drinking happened earliest, that there's this variant. And the one thing I just want to go back and explain is when I was showing you the picture, I showed you it in one direction, but you're actually looking on both sides. Look to the left, look to the right, and see how correlated things are. And so here's the variant in Europe that's under pressure. And you can look to the left, look to the right. Everyone is very, very correlated, suggesting that that variant happened very recently. And then this is in comparison to another common variant, for example, in Africa, where you can see a lot more breakdown. And so it suggested that lactose happened recently in the past and spread quickly through Europe. And we also started looking at other variants that we knew something about. Here's a variant, it's CCR5. In fact, one of the first studies of natural selection in humans, or most famous studies of natural selection in humans, was at a locus that protected from HIV at CCR5. And they thought that that variant that protects from HIV happened in the last 700 years and proposed that maybe the plague was driving its rise in the prevalence in the population. And we went back to explore that with some of our methods to see what we found. And in fact, here's the resistance variant, and it lies on a long haplotype, suggesting that it's a fairly recent age. And then we started looking at other variants at the locus. And here's the non-selected variant, but it also lies on sort of a long haplotype as well. And these are not that common. I mean, so lactase had gotten to 80%. This had gotten to about 8% in European populations, so not a high prevalence. And what we found actually is that, in fact, in light of new data, once we started getting data from the whole genome, we saw that in light of new data, that variant is actually not that unusual. So things that are, it's rare in prevalence, so things that prevalence lie on long haplotypes often, especially because there's a historical contraction of European populations, that kind of thing happens. So it shifted in there. I said this was a very, very popularly known story, and so it made us have to rethink what we were looking at, and it made it clear that we needed data from the whole genome to get a sense of what was real and what wasn't. And luckily, that data came. So we have a new era of whole genome data where that became available. And one of the biggest, there are a lot of different data sets that became available, but one of the biggest ones, and the ones that I got to be a part of, was the International HapMap project. And for that, it was 8 centers in 6 different countries that got together to study 4 million different variants across the human genome in 3 continental populations from Europe, Asia, and Africa. And so so we worked together as a collaboration, and I was part of the analysis team studying selection. So I talked to you a little more detail about the type of work that I do, just because I know it's sort of, I'm more comfortable with it, and a lot of the things that will show you are from that kind of method. But when we worked with this global project, we used several different methods. So and I just want to go through them with you to show you that there's actually just a few set of things that you are going to find with natural selection. So natural selection is the rise in prevalence of this variant from low prevalence to high prevalence. But it leaves certain different signatures that we're finding. So population geneticists are just trying to find, well, what else could it do? What are the different things? But it's based on that same principle I told you about. Before, there were no copies, and then there was one copy, and then suddenly it spreads and becomes very, very prevalent. On a DNA level, this is what it looks like. So before, it doesn't look exactly like this. It's not purple and red, but so before this is, let's say there's 5 variants I'm just showing you a schematic of, and the red one is the one that is the selected mutation, that selected variant. And here's some other variants on that chromosome. And so because that reshuffling process I was telling you about happens pretty slowly, recombination, if the selective event happens very, very quickly, there won't be time for reshuffling. So not just that variant, but every variant on that chromosome will go up very quickly in prevalence. So you can see that that's the chromosome in the middle, and so every variant on that chromosome goes up. See the purples that come with it. And so what will happen is it'll leave these signatures in your DNA. So what are the different kinds of signatures you might be able to see? Just by looking at this cartoon, you could actually find. Well, one is something called low diversity many rare variants. So as that chromosome goes up in the population, everything else does too. And so all the other variants start getting driven out. So you can see on this, that 2 of these variants just disappear because the other form of it are pushed out of the population. So in some cases, that variant just disappears completely. You can't see it anymore. And if it goes to 100% prevalence, that chromosome, basically all the variants disappear. You no longer can see another form of it, so you don't even know there's a variant there. So we see a dip in diversity in that region, genetic diversity. And whether it goes to 100% or really high prevalence in the population, you're going to see a dip in diversity and many rare variants existing in that region of the genome. So remember I told you that it takes a long time for a new variant to get up in prevalence in the population? So we would just say the new variant is what we call the non-ancestral state. So before there was a mutation, everybody in the population was exactly the same at this position, and you don't really see any variation. Then a mutation occurs, creating the non-ancestral variant, in what we call a derived state. And in this case, the purple is the schematic of the derived state. And because it takes this very long time for that new variant to rise in population, the derived state will normally be at very low prevalence. But as that chromosome goes up in prevalence in the population, pulling with it everything else, not just this one, which is the derived state, the selected variant that's the derived state, but other ones on that chromosome will also go up in prevalence. And so we see this high frequency of derived alleles. And then we see those long-range haplotypes that I was telling you about before, so I won't dwell on that. And then the last signal that we were using to mine the data set from the whole genome was population differentiation. So I had said that natural selection is a process by which we adapt to an environment. So if that happens to be a local environment, that's going to drive variation very differently in different populations, right? So here's an example of something that protects from Plasmodium vivax malaria, where the white suggests that the variant is completely absent outside of Africa, but it rises to about 100% in certain parts of Africa where Plasmodium vivax was once endemic. And so human populations are actually quite similar. They're quite related, and this kind of a dramatic difference between populations is unusual. And so but it's something that can happen when there's selective pressures in one population and not another. So these are the types of signals of selection. And so now we developed software to be able to analyze the whole genome and went through, which is available on the web and lets anybody really browse through the data looking for things. And we looked to see what we found. So all of the stuff that we're doing is we're just looking at the most extreme things in the genome to see what we find. And we want to know whether or not these things are real. What's nice is that one of the first things we found, and one of the strongest things we found, is something we already knew a lot about biologically, which was that lactose tolerance. And in fact, actually, that huge chunk where this thing is correlated over a long, long distance and gotten to high prevalence is, in fact, very unusual when you look at the whole genome. And so this is a picture of chromosome 2, and this is the significance of the test that we find. We see a very, very strong signal. So in fact, lactose tolerance is, once we look at the whole genome, one of the strongest signals of selection. Now here's a picture of just some of the things that we found in Africa. Well, I told you that one of the things we knew about was we found CD40 ligand, we knew about sickle cell. Well, in fact, when you actually look at the whole genome, not just those variants, but many other variants that protect from malaria are in the top 100 candidates that you would find. So CD40 ligand, the HLA, something called CD36, many genes that have protection or effects in malaria were under selection. So we can see what a strong pressure that was. And what are some of the other things that we find? Well, here's one of the top candidates in Africa, and it's a gene called LARGE, and it's on chromosome 22, and you can see this really strong peak suggesting strong selection. So we looked at LARGE, and we wanted to know what it did. It's as it says, a very large gene. So the whole signal of selection is encapsulated within that gene. So we just did a PubMed search. We found this. This is that signal. It's a 35% variant in West Africa, in Nigeria, that seems to be under selection. And so in a PubMed search of LARGE, which is difficult to do because LARGE comes up on everything, so try to do a Google search on LARGE, you won't get. But we did, in fact, finally, actually, find this beautiful paper that talks about how LARGE is critical for the binding of arenaviruses. So without the gene LARGE, arenaviruses can't get in because it does an important modification of the gene that's the entry port. And so what are some arenaviruses? Well, I told you that the signal of selection was in Nigeria, and so what was interesting is that the most important arenavirus is Lassa, named after Lassa, Nigeria, where there's a virus. It's a virus that causes a hemorrhagic fever, and it kills about 5,000 to 10,000 people a year. So I'm looking for natural selection. I'm thinking for major shifts in the population. So something that kills such a small fraction of people you wouldn't really think to be under natural selection. But what's interesting is what we're looking at are events that happened in the last thousands of years. So in fact, actually, what we found was that LARGE only kills a small fraction of the population, but that about 25% of the population are infected. So, and in some parts of West Africa, 50% are. So, and most of these people get this virus and only get a small fever. And so they have an immunity to it. So this is very interesting, and it suggests that what we might be finding is a mechanism by which a past strong selective pressure has now been changed by a beneficial variant arising to it. And so that's what we're studying now. And I told you before, we found a lot of genes that protect from malaria that were involved, and so there's a list of them here. And this is one we didn't know about. We didn't know about the selective pressure occurring at it. And what was nice is that we see the selective event where we see the strong correlation, and we see that it localizes in on what we call a nonsense mutation, something that knocks out this gene CD36. And we know that this is very critical for how the parasite hides itself away from our immune system. And we were able to localize in on a functional variant that was on this haplotype, suggesting a selection. So in these genome-wide surveys that we did, we found some of the things we already knew about. So before the genome era, we had already found some things like lactose tolerance, like some of the things that protect from malaria. But the vast majority of what we're finding are absolutely new. And the challenge we now have is to sift through all these regions and find the things that are driving selection. And so what are the first things you can do? Well, these are large regions that we're looking at. We're looking at correlations that occur over vast areas. So it's not so easy, actually. And some of these areas contain many, many genes, and some of them are with many different interesting candidates. And some of the regions contain no genes at all. So one of the most interesting candidates is this chromosome desert, a gene desert on chromosome 4. So 3 megabases with no genes at all that seems to be under very, very strong selective pressure. But what's interesting about that region is it's heavily associated with obesity. So this may be a regulatory region that is under selection. So how do we localize in on the selected variant? So I told you before that the thing is this long haplotype is this long region where everything is connected back and forth to each other. So it's very hard to say which one's the actor. Every variant across that whole haplotype has a signal of selection. But the thing is that what we're looking for when we look for selections, we're looking for a young variant that rapidly rose to high prevalence. So we'd expect that that variant happened sometime within the last 6 million years, right? We expect actually it happened within the last 10,000 or 20,000 years. But the suggestion is that it's not something that happened before the split between chimps and humans. So we can look for the non-ancestral variant and say, on that chromosome that we're looking at, how many of these variants are the non-ancestral state that was driven up? And that's only a fraction of the things that are on that chromosome. We also know that selection is a local adaptation to the environment. So we can look for populations where we identify selection and those where we don't, and see whether or not we see that that variant has been differentiated between those populations. And we can overlay this and see whether or not the data kind of converges on a particular variant. And one of the most important things that we expect for the selective thing is that it affects your physical trait. It has some sort of a function that you're going to see, right? So that's actually probably the most important thing, right? For something to cause selection, it has to have an effect of some sort. But it's actually one of the hardest things to know because we don't have a really good catalog of what variants in the human genome are important or not. But basically, as a last sort of pass, we took all of the things that we know, kind of different kinds of motifs and factors that we know to be functionally important, and we said, are any of these things that we're looking at one of those? And so we did that, and we applied it to our regions. So like I said, it's very hard to figure out what's the actor, what's the variant that's important, because these regions are so large. So there's 700,000 positions large in this example. But the thing about this example is we actually know something about it. There's an amino acid change in a particular gene called SLC24A5 that has already been shown in zebrafish to be critical for pigment. And in Europe, in Europeans, it's that variant, this particular amino acid change, drives pigment differences between Europeans and other populations. And so we said, well, let's use this kind of process and see how it would work on something we already know. Would we find the variant we're looking at? So in this region, there's 867 variations that all have that signal of long haplotype, so a huge number. Only 233 of those are the non-ancestral or derived variants. Only 12 of them are differentiated, so actually very few of them are really driven differences between populations. And we say, how many of those is it that the derived variant is the one that's been pushed out in that population? And it's only 5. And then we overlay that last piece of evidence as to whether or not we think it's functional. And in fact, we actually localize in from all of the 867 variants to only one likely candidate, this very strong signal at threonine to alanine in that gene. So this kind of a process actually, at this gene and at lactose, where we already knew something about, localized in on the variant we were interested in. So we did the same thing with our best candidate in Asia, so something we didn't really know anything about yet. And same kind of thing. Now there's even more variants, and it's a longer region. And there's 933 things. And we sift down and we get back to just 5 things that are those top candidates that seem to be driving selection. And only one of them is functional. And it happens to be amino acid change in the main what we call the death domain of this gene EDAR. So it seems to be a functionally important change in the gene EDAR. And so we looked to see, what do we know about EDAR? Well, in fact, we actually recognized it immediately and were excited by it because David Kingsley at Stanford, made this pathway very famous because he identified that in stickleback fish, this same pathway was under repeated selection. For scale, so in fish, this pathway drives the production of scale. And in freshwater fish, they were losing their scale repeatedly for many reasons. And we saw natural selection acting on this pathway. Well, here, in fact, actually, we see strong selection in Asia acting on the same pathway, but it affects a completely different physical trait in humans, right? Still on the ectoderm, on your external surface. It regulates hair, sweat, and teeth. And in fact, so mutations in humans, we know a lot about its function because in humans, mutations in it cause sparse hair, a lack of sweat glands, and malformation of teeth. And so we looked in, so when we look at this, we're trying to figure out what are the clues? How do we know what this is driving? Well, one thing we can do is just look at the global population and say, well, where does this variant exist, and does this lead us to any clues? We can see in the Pima Indians, it's 100% of the people have this variant. In parts of northern China, 100% of people have this variant. But in places like Japan and Thailand, in fact, it's variable. So only a fraction of the people have the variant. And so we started to look at the anthropological record and see what are the differences between Pima Indians and Chinese and Japanese with hair, sweat, and teeth. And in fact, actually, there's a very, very rich historical record about teeth. And in fact, there's something called, sort of predating PC, called the Oriental cusp, where it's a type of tooth shoveling that is existent in populations, and the frequency of that, the prevalence of that tracks with this trait. And also we know that the Japanese, which is one of the populations where it's more mixed, are known for having a lot more hair than other Asian populations like Chinese and Korean, because they're founded by a population in part that are known for excessive hair growth called the Ainu. And so these are clues, and we're now actually working biologically to figure out what this does. And there are many more intriguing candidates. So, I talked to you a little bit about the fact that we found things important possibly in a hemorrhagic fever, possibly in hair and growth, and then possibly in pigment. But when we look at our list of candidates, in fact, not just that gene large, but other genes that are important in that hemorrhagic fever and how it binds are also under selection. Not just EDAR, the gene that I was just talking about, but other genes in that same pathway are driven in evolution, not just in Asians, but in other human populations. And also in pigment, we see many, many genes involved in pigment that are also under selection. So what we're seeing here is that we can see these different principles driving human evolution, and in fact, they may be whole pathways that are being driven in the same way. And so where are we now? We have these new tools to study human evolution and the new data sets. We're finding that by looking at our top regions that we know so far, we know that infectious disease is important, changing environment, domestication of plants and animals, and these are major things driving human evolution. But there are all these novel candidates that will give us whole new ideas of what are the principles driving human evolution that we have to follow up, and that's our next challenge. But one of the things that I became very impressed by, and with my interest in infectious disease, is how strong of a force malaria is. It's quite, I have so much respect for this parasite. It's been around for 100,000 years in the human population, probably much, much longer. There's no reason not to believe that it's actually been in human populations since the split between chimps and humans 6 million years ago. And so this is a parasite that has stuck with us for such a long period of time and never gone away, yet still remains so pathogenic. So you would think that if it wants to, it would want to fly under the radar, right? It would want to sort of hide with us and not bother us. But in fact, the fact that it's still killing 2 million people a year, it's still having such an impact on our genome, suggests that it has never sort of stopped being an adversary. And so how is it staying around? How is it evading our immune system? And now how is it evading the drugs that we develop? And so that's the next thing that we're looking at. And I'm just going to tell you a little bit about what we found so far, because this is a very early stage in the type of work that we're doing. But where we want to go is not just understand how we're evolving, but also the pathogens that affect us, to get a sense of how we can fight sort of these adversaries. So the genome of Plasmodium falciparum malaria has been sequenced, but very little is known so far up till about a year ago, about genetic variation in the parasite. So we were sort of in those same dark ages that we were within humans, where we only had about 100 different variations across the 24 million variants in the human genome, in the malaria genome. And so we wanted to focus in on that and find these variants and begin to use these kinds of methods. And so we took a set of parasites from around the world, working with, and I worked with Diane Worth at the School of Public Health and Sarah Volkman. And we got many, many parasites from around the world, and we started sequencing, looking for these kinds of variations. And we found in our first stage 47,000 variations across the malaria genome. Using many, many different types of data sets, really getting a sense of what exists. And so just in this very first phase of the project, we found some interesting things. One of the things that we did is, so there are different kinds of genes, different kinds of proteins in any genome. And we can classify those differently. So we can say, for the parasite, what are things expressed on the cell surface? What are things that drive metabolism? We can take those sets of genes and group them together. And we can look for things that, how many of those genes don't have any diversity at all? How many of those have a lot of diversity? And what we find is that in the classes of genes that have things with a lot of diversity, most of those are things that are expressed on the cell surface. So they're things that are in fact interacting with our immune system, where the parasite wants to constantly change to hide itself from the immune system. Whereas things that don't change hardly at all, that have many more genes that have no diversity, are things that are important with metabolism, things that just basically hold down the ship. But what was nice is already looking at this, so we can get the sense of this is how the genome works, and it makes sense. But it also points us to some interesting biological things. So things that expressed on the cell surface that interact with our immune system, that don't have any diversity at all, are wonderful vaccine targets, right? Because the problem with most malaria vaccines is that you develop one, you deploy it, and then it just mutates and it's no longer working. So we're going to focus in on those genes that have no diversity, that are expressed on the cell surface for vaccine targets. One of the other things that we find is when we think about malaria, we think about the parasite, most people just think of it as one strain. You don't really think about the diversity that exists. But in fact, actually, the malaria parasite is infinitely diverse. And if you look at parasites from Asia, America, the Africas, they're very, very different. And their pathogenesis, what they might do to us, are very different as well. So getting a sense of all the different parasites that exist and what they're doing is important. And finally, we can begin to deploy these tests to look for natural selection, finding interesting biological things. So again, we always go back to these same sort of schematics of before selection and after selection, what that would look like. Well, in the case of drug resistance, you can think of the before population as a drug-sensitive set of strains, right? And then after you put in a drug, you create a selective pressure, and then you get drug-resistant strains. So if we think about those as the before and after, we can look for regions of the genome where sensitive strains have a lot of diversity and resistant strains suddenly have a huge dip in diversity. And that can point us to loci that are important in drug resistance. And so we did that with the data that we had generated so far, which is a very preliminary data set. And we found 3 different loci that are associated with chloroquine resistance. And one of them was the known chloroquine resistance transporter that is highly associated with drug resistance, chloroquine resistance in the parasite. So this type of method did find something that we knew about. But we also did it, but in looking at it, there are also many things that we didn't know about that we were finding by using these methods. And here's a very, very strong signal that we find on chromosome 13 for pyrimethamine resistance, which is one of the other commonly used drugs. So this type of a method is actually allowing us to quickly pinpoint regions of the malaria genome that are changing towards drugs. And the last kind of thing is that we can actually not just employ the diversity type of test, but all sorts of tests at the malaria genome. And I had said before that we use this length of haplotype. Well, in fact, because the recombination is very high in the malaria genome, the length of haplotype is quite short. So these long haplotypes stand out. And so when we actually look through the malaria genome, we see a lot of these really interesting signals where we see a new allele that is highly correlated over long distances, suggesting selection is acting. So in the next phase of this project, we're really trying to develop this map of diversity in the malaria genome. So this is a very early stage where we're identifying all the variations that exist. We're finding ways to measure those in patient populations. And then we're doing all these kinds of things to look at the populations, look how different they are, identify drug resistance, and create tools that we can actually also deploy to the malaria community in the field. So diagnostics that can be used right on site in the places that need them. And one of the big projects that we work with to do this, and we're starting to, we have a lot of wonderful sites in Senegal and elsewhere, but we're also working with something called the Millennium Village Project, which is run out of the UN. That right now is in actually 11 countries and growing, where they're trying to bring these villages out of poverty, and they're working very closely with them. And we're helping them with their diagnostics and with their sample collections. So in our studies of malaria, right now, as of today, we're at 120,000 variations across the malaria genome. And we've already begun to identify these sites that are under natural selection and identify the diversity that exists, and finding these drug-resistant loci to work towards. And we're continuing to expand those efforts, working with the worldwide malaria community, both in the Millennium Villages and elsewhere. So I have a little video that I'll show you, depending on time. I don't actually have how we're doing on time, but I'll show you a little video, because it's nice sitting here in our lab. In Boston, you don't get sort of a sense of the global implications of the work that you do. So we actually get to go out to Africa on a regular basis and interact with those populations, which is highly rewarding. But in summary, for what we've done so far, or what I've discussed today, we've gone over some of the methods for studying selection, and now basically all the tools in our toolkit for the most part. So you're experts now. So you have some of glimpses of the kinds of evidence of natural selection that we found in humans. And you have a first glance at what we can do in the parasite and what we're aiming to do. And really, we're just wanting to continue those studies to investigate natural selection in humans and pathogens and how they interact. And so just to bring it back to where we were before, to give you a sense of the global view of where we're at. Like I said before, even before Darwin and Wallace, there are many, many other thinkers that brought us to the idea that species are evolving and adapting to their environment. But in 1858, we got the hypothesis which drives that, the idea of natural selection. And in 1871, humans were brought to the forefront in that discussion because of the descent of man. But it wasn't until 70 years later where we had any idea, of anything, any trait in the human populations that were in fact evolving, and proof of the connection that exists between malaria and sickle cell. And since that time, people have been doing these mining expeditions, looking throughout the genome, finding new regions of selection, and some of those with new data actually disappeared. So we're really sort of going through it slowly. But now in 2004, 2005, with data from the whole genome, we can find a rich number of regions that are under selection. And those things can help us study the forces that have shaped our species and other species that we interact with, and also can provide important insights into human health. And so with that, I just want to give my acknowledgments. This is a list that's very, very short. And so the way science is now, you work with so many people and it's such broad communities. But here's just sort of like a hot list of some of the people I really want to thank. Most importantly is Eric Lander, my advisor. He's actually been my advisor for 12 years and is an extraordinary mentor and an extraordinary scientist. And Diane Worth, who is a great light in malaria research and who's brought me into that field. And a rich number of universities and funding sources and consortiums and institutions that have worked with me. So if you want, so bear with me. Here's a
that we have many collaborations, but this collaboration is one of the best. I will not say the best, but that's what I'm thinking.
So that's just to acknowledge the most important people, which is the people that we work with throughout the world. So, thanks. So, any questions? Or? Yeah?
When you go to a population to see if there's a variant, how many subjects do you have to do a sequencing on to find out?
So one of the neatest things about this kind of approach of studying natural selection is how powerful it is. So in order to do an association study where you take individuals who have malaria and those who don't, and find out whether or not a variant is protective, you might need thousands of patients, very, very carefully clinically diagnosed and measured for the level of disease to study this. But in fact, actually, the fact that the type of approach we're using is using generation after generation of small effects, right? Small protective effects that allows this thing to go from one person in the population to many people in the population. It makes it so it's almost like financial compounding the bank, where it creates this whopping signal sometime down the line. So the types of signals I'm finding you could actually go, and I mean, at the strongest signals, you could take like 30 people and find this type of a signal. But mostly, when I was saying before, I work with this consortium of individuals, where it's this consortium that studies 90 individuals from each population, and we found so many of these signals. So you could do it with a very small set of people and find these strong signals. I'll also be out there. Yeah, go ahead.
You discussed the prevalence of something over time. Do you have any way of measuring how the prevalence changes? Is it a linear growth?
How the prevalence changes? So the question was that we talk about the prevalence that occurs over time. Is there a way of measuring how the prevalence changes? Well, we can get a sense of, we can predict how long ago something occurred, right? So, but it's hard, obviously, to track what's happened in the past to now. But one of the things that's interesting is, so for lactose tolerance, the prediction based on how much variation exists around that region is that the thing happened within the last 10,000 years, so 5 to 10,000 years. And so what's nice is that tracks with the introduction of cattle, so it gives us a good sense that this might well be right. And now with data from so our ancient DNA technologies have gotten much, much better. Our ability to go back to bones from a long time ago and see what existed then. And so a few studies have happened where they've looked at skeletons from the last 10,000 years ago, and they saw that, in fact, nobody in those populations had that variant. So it looks like, I mean, that's only one sampling, and they'd have to do much more of that to prove that that didn't exist. But we can see that actually 10,000 years ago, nobody had that variant, and now we do. But for the most part, it's hard to think about what are the fluctuations that occurred. What did it look like 5,000 years ago? What it looked like. You really actually probably need samples from each of those time points to see what's changed. The great thing about malaria is that, in fact, actually, their generations are so quick, right? We are looking at something every 25 years a shift happens. But with malaria, every few months, there's a new generation emerging. So in fact, actually, right now, we're going back to the parasite samples that we had back in the 60s before, chloroquine resistance arose, and we actually are able to we have almost a fossil record of malaria and what's going on. And we can track real time in the field as we introduce vaccines and drugs how it's shaping and shifting. Sorry, the lights are right on me. Yeah. Oh yeah, go ahead.
How do you protect yourself during these visits?
How do I protect myself during the visits? Yeah, well, I've been using Malarone, so, but obviously drug resistance is rising everywhere, so you take a risk when you go. But just this year I went to Kenya and I went to Senegal. And in the next years, we're probably going to go to many more sites, including Cambodia. And so you just look, what's interesting is actually the drug resistance in some populations, some drugs are resistant where they're not in others. So there are these local changes depending on what drugs they use. So you just sort of pay attention to the CDC and WHO and figure out which ones are the best to use. Yep.
Do you see traits that co-evolve?
Do we see traits that co-evolve? Well, so it's interesting. We don't. We're just really at sort of the tip of an iceberg. We don't exactly know. So it's actually interesting, this whole idea that when I started looking at our lists, we found that like it wasn't just the one gene in hair, but all these other ones, one genes in pigments, and all these other ones. We're now beginning to get a sense of that. So we still are just trying to figure out what is under selection, what are the pressures, and then we can start looking at that. One interesting example of coevolution, especially when you start thinking, people are always asking me, you're studying the parasite, you're studying humans, do you see something that's happening between them? Do you see coevolution happening there? And I don't really know yet, because I haven't looked enough at the malaria parasite. But that example that I had showed you before, I had said there's the CD36, this knockout of this gene that is important in malaria resistance. We see a strong signal of selection occurring there. What's really interesting is that we see the signal of selection at this knockout, but that knockout actually causes susceptibility to malaria. So we know it's important in pathogenesis, but it's the thing that would make you to get malaria more is actually the thing that seems to be beneficial. So that's counterintuitive. And so the things that could be driving that is one, it could just be one or both could be wrong results, right? So you can't ever rule, you want to try to rule that out. But the other one is that actually it may cause you to get malaria more often, but maybe not die of it, maybe not affect your longevity. So it's this very interesting interaction that happens it's how the pathogen hides from your immune system, and there's all this other stuff going on. But another hypothesis could be that in fact, what once was actually protective is now no longer, right? Because what we're looking at when we look at selections, we're looking at something backward in time. But now today, it causes susceptibility. So we actually know what that receptor binds to in the parasite, and we're studying that region as well to see whether or not an interaction has taken place.
Well, please join us for a reception that's following in the lobby, and join me in thanking Pardis for her.
Thanks so much for your time.